Okay. You are muted to me. I can hear you. You are muted to me, Natasha.
All right, Stefani. So
now yes.
You can hear me? Sorry about that. Yes. So our next speaker is Stefani Altenhofen, who is going to present Dichlorvos, which alters morphological parameters and behavior in zebrafish. Back to you, Stefani.
Okay, hello everyone. My name is Stefani Altenhofen, and I'm a postdoc fellow at Pontifícia Católica University of Rio Grande do Sul, located in Brazil. As Natasha said, I will present the work entitled Dichlorvos alters morphological parameters and behavior in zebrafish. The central nervous system has sophisticated functions, such as the ability to control a lot of activities. The primitive human brain develops into a complex organ during prenatal life. To achieve its full development, connections must be made along precise paths aimed to communication between cells. The time for this process to occur must be rigid and controlled in order to obtain the correct sequence and timing in each stage of development. If the development process of the immature nervous system are impaired, the effects can be long-lasting and permanent, leading to teratogenicity, deficits in mental capacity, and behavior disorders. In social aspects, the consequences of neurotoxicity include the increased probability of school failure, decreased economic productivity, and increased risk of antisocial behavior and sociopaths. Neurotoxic agents can cause brain damage, especially if the exposure occurs during the early developmental period. Among the documented agents are industrial chemicals, such as pesticides, organic solvents, or metals, which are commonly found in air, food, water, and soil. Agriculture has been practiced by humanity for over 10,000 years. However, the intensive use of agrochemicals to control crop disease and pests has existed just for over half a century. The main best known class of agricultural pesticides comprises organophosphate, a family of agrochemicals developed after World War II, chemically derived from phosphoric acid. To induce neurotoxicity, this class acts by inhibiting acetylcholinesterases, causing accumulation of acetylcholine in the synaptic cleft and the hyperstimulation of the cholinergic receptors, triggering an acute cholinergic crisis able to induce convulsions, alteration in the ventilation, metabolic imbalance, and death. Dichlorvos is a highly toxic insecticide belonging to the organophosphate family, considered one of the most used in agriculture. It was synthesized in late of 1940s, and it is used in the control of insects in farming. This organophosphate causes intoxication through of inhalation, cutaneous absorption, or injection, being persistent of degradation under natural environmental conditions, and may be deposited in the fruit or water. An important concern related to the frequent application of pesticides, such as organophosphates, is their accumulation in groundwater, causing ecotoxicological effects, especially here in Brazil, where the laws regarding pesticides are very soft. Studies using model organisms such as zebrafish to assess the toxicology of chemicals present in aquatic ecosystems can provide data for environmental risk assessment. In addition, late evaluation of exposure to organophosphates at the early stages of development is important to elucidate the risks that these agents may bring in the future. So in this study, it was evaluated the toxicological effects of Dichlorvos exposure during zebrafish development by the analysis of morphological and cardiac alterations in zebrafish larvae, acute toxicity by LC50, analysis of exploratory and aversive behavior in zebrafish larvae, and locomotor and social behavior in adult zebrafish. For treatment, embryos were placed in petri dishes and subjected to water for control group, or Dichlorvos at concentrations of 1, 5, and 10 milligrams per liter, since 1 hour post-fertilization to 7 days post-fertilization. After the treatment, the animals were placed in 3-liter water tank until 120 days post-fertilization. Here in the experimental timeline, we can see that the morphology and heartbeat rate experiments were performed at 7 days post-fertilization. The assessment of exploratory and aversive behavior were performed at 7 and 14 days post-fertilization. Locomotor activity was verified at 30, 70, and 120 days post-fertilization, and social behaviors were analyzed only at 70 and 120 days post-fertilization over social preference and aggression protocols. First of all, we estimated the LC50 value. So this analysis was evaluated during exposure and based on the mortality of a total of 100 animals. Here in table, the results show that LC50 values between 24 to 168 hours were 62.45 to 21.51 milligrams per liter, respectively. So based on this analysis, we choose the concentration to continue the study. To morphology analysis, we performed at 7 days post-fertilization over the evaluation of body length, ocular distance, and surface area of the eyes by Nikon software. Body length was measured by the distance from the larval mouth to the pigmented tip of the tail. The ocular distance was evaluated by the distance between the inner edge of the two eyes. And the size of the eyes was determined by measuring the surface area of one eye of each larva. Usually we choose the right eye. The results present of figure B showed that the Dichlorvos exposure caused a reduction in larva body length in all concentrations tested. However, there were no differences in ocular distance between the control and Dichlorvos treated groups. On the other hand, the figure D shows that the surface area of the eyes also was reduced at 5 and 10 milligrams per liter, but there was no difference in concentration of 1 milligrams per liter. The analysis of the cardiac effects of Dichlorvos exposure was performed by heartbeat rate. The measure occurred at 7 days post-fertilization under a stereo microscope. The animals were placed in petri dishes with water, and their heart rate was monitored by 10 seconds and multiplied by 6. Discount was done in triplicate for each animal. And the results demonstrated that a decrease in heartbeat rate of larvae exposed to Dichlorvos at all concentrations analyzed. The exploratory behavior of the larva was evaluated for 5 minutes at 7 and 14 days post-fertilization by Animase software. Each larva was individually placed in a cell culture 24 well plate in a design protocol that virtually divided each well in inside and outside area. Animals exposed to Dichlorvos at 1, 5, and 10 milligrams per liter displayed reduced travel distances and mean speed at 7 and 14 days post-fertilization, as you can see in figure A and B. Moreover, the figure C shows that the animals exposed to Dichlorvos at concentration of 1 and 5 milligrams per liter showed no significant difference in time immobile at both ages, 7 and 14. However, 10 milligrams per liter reduced this parameter in both ages. The time spent outside the well area measured showed no significant difference in both 7 and 14 days post-fertilization between treated and control groups. After the exploratory evaluation, the animals were placed in 6 well plates, 5 larvae per well, over a LCD monitor to measuring cognitive ability and avoidance response to a visual stimulus. This visual stimulus was a riding bouncing ball. The analysis was performed over the number of larvae that avoided this stimulus. There was a decrease in the percentage of animals with escaping responses from the aversive stimulus in all Dichlorvos concentrations tested at 7 days post-fertilization. At 14 days post-fertilization, there was also a reduced percentage of animals that escaped from stimulus at Dichlorvos concentrations of 5 and 10 milligrams per liter. The locomotor behavior of the adult animals were evaluated for 5 minutes at 30, 70, and 120 days post-fertilization, also by Animase software. Each adult animal was individually placed in the test tank that was virtually divided into equal sections, upper and bottom zone. Here in figure A and B, are the findings regarding distance traveled and mean speed. The animals exposed to the insecticide at all concentrations displayed decreased distance traveled and mean speed at 30 days post-fertilization. However, only the higher Dichlorvos concentrations were able to reduce these parameters at 70 days post-fertilization. At 120 days post-fertilization, there was no significant difference between treated animals and control group at distance traveled and mean speed measures. And the time mobile and the time spent in bottle zone shows at figure C and D did not present significant changes at any of the ages analyzed. Social preference and aggression were assessed only at 70 and 120 days post-fertilization, since the social and aggressive behavior of zebrafish are not completely developed at the other ages. So the protocol of social preference consists of 3 tanks, one empty, one central called experimental, and one with 15 animals called stimulus. To quantify fish preference, the amount time of the experimental fish spent on the stimulus zone was measured. The aggression analysis was performed using the mirror test. Each fish was placed in an experimental tank test, and the mirror were placed at the side of the tank at an angle of 22.5 degrees. The aggression behavior was quantified over the amount time of the experimental fish spent close to mirror. We also evaluated the number of bites and chasing movements against the mirror image as an aggression parameter. The results shown that the Dichlorvos exposure during initial phases of development was not able to alter the social parameters in adult stage. So in summary, Dichlorvos exposure was able to alter morphological, cardiac, exploratory, and cognitive parameters in zebrafish larvae at 7 days post-fertilization. Moreover, this pesticide also alters the exploratory and cognitive behaviors at 14 days post-fertilization. Already in adulthood, the exposure of Dichlorvos remains causing changes at 30 and 70 days post-fertilization, reducing locomotive parameters, of the animals exposed in early stages of development. However, at 120 days post-fertilization, all locomotor parameters of the exposed animals are similar to the control group. So taken together, these results suggest that Dichlorvos caused significant changes in central nervous system. On the other hand, an important point of our findings is the fact that after the Dichlorvos removal, the exposed animals develop an adaptive mechanism, recovering the locomotor behavior at adult stage. The possible response for a decrease in the Dichlorvos neurotoxicity could be related with neurogenesis and synaptic plasticity. However, despite the recovery, locomotor impairment over the period present in this study can be fatal to the animals in life in an environmental infected by this pesticide and other pesticides that need to be studied, showing up as a warning for agrochemical use. So I would like to thank and congratulate the organizing committee, and I would like to thank my university, the Brazilian founding agencies, and my lab colleagues. Thank you all.
Thank you very much, Stefani, for your talk. So Alexi has one question for you. It looks like Dichlorvos is altering exploratory behavior and social ones. Could it be linked to the vision impairment you observed during the development phase?
Maybe. You see visual, yes?
Yeah.
Maybe if we analyze the optomotor analysis, we can provide this answer. But with these results, I think that I can see that. Yeah.
All right. So we are still waiting for questions on the talk, but you are already congratulations. I have one question, which is a really naive one because I'm not studying fish at all. But it is my understanding that zebrafish is a model species and that very little is known about the ecology of this species. So how can you expand the effect of Dichlorvos to other species?
Maybe zebrafish is a little small fish, and when you study this animal in a big scenario of bioaccumulation, and we can think how ways the toxication that zebrafish have can pass it to other species, especially in thinking this bioaccumulation ways. I don't know if I respond to your question.
Yeah, kind of, yeah.
Yeah.
Okay. So I will wait a few minutes more and maybe questions will pop up. If not, they will ask you the questions directly on the Discord server if it's fine with you.
Yeah.
Sorry about the beginning of the video, by the way. That was my fault. So do you know about the geographical distribution of zebrafish or not at all?
What?
The geographical distribution of zebrafish.
It is a small fish from India, so I don't know the distribution in entire world. I don't know this.
Okay, you have one question from Patty Hens. She says, "My talk. What experiment would you do to answer the question of Alexei?"
I think that it was the optomotor of the zebrafish. It is a experiment when you put the larva in a LCD monitor too, and lines were passed, and the animal needs to in encounter of the lines. So we saw the visual presence of the zebrafish.
All right. All right, then. Thank you very much,